Okay, well, thank you. As the other speakers, I would like to thank Professor Demenko and Professor Bard for having me here. I am kind of learning quite a few things, basic concepts about climate, and I think that after I present the talk, there will be room for discussion on how can we fit genetic data and climate inferences. So some of you have asked me this question. The best thing to do if we want to know what North African were doing during the Holocene would be go and sequence people from the Holocene. But unfortunately, this is not always possible. Sometimes because we don't have, unfortunately, a good fossil record. It also depends on how good the fossils have been preserved. There is the high risk of contamination, and it's also very expensive. But luckily, we can use present-day data, genetic markers, to answer some questions that related with human demography. So, for instance, whether populations grew, whether there was a reduction in population size, or whether there was migration that we call gene flow between populations. So, in order to give a bit of background, I will start far away from the Holocene, and hopefully we can have some discussion on that because there are also very interesting results related with climate, and we will start between 60 and 40,000 years ago. So what happened there was that modern humans went out of Africa. It is kind of established now that probably modern humans also went out of Africa before, but that time was really relevant because it was like the time where afterwards they expanded towards the whole globe, and also that entailed the extinction of other hominin species like Neanderthal, Homo erectus, Homo floresiensis, and some others. So to now, basically, most of the people agree that the origin of modern humans was in South Africa, but a few years ago, there was a study that discovered some human remains that claimed to be modern in North Africa that dated as far back as 100,000 years ago. So there was a new hypothesis that was establishing that, well, maybe North Africa was actually the out of Africa, like the population that went out of Africa. Why not? So far it's only a hypothesis, but it's kind of interesting in the background. So maybe this is too simplistic for climate experts, but this is a diagram that is showing that basically at that time period, that it's between 60 and 40,000 years ago that I have highlighted in green, the climate was basically much drier and cooler than what it is now. So here in the x-axis, we have the mean temperature, the present mean temperature. Sorry, in the y-axis, we have the present mean temperature and differences from that present temperature. And in the x-axis, we have the time scale. So you can see that it was not only drier and cooler, but it was also there were a lot of fluctuations. So this is interesting because the former migrations out of Africa were actually related with other mammal migrations. So it was a kind of related movement. But that time, it was only humans who went out of Africa. And trying to understand why did they do that, it's not clear at all, at least not to my knowledge. And if you have some feedback or thoughts on that, it would be great. So this is also I try to represent what was the vegetation at that period, and this is basically the red square. Is there a pointer that? Oh, wait, here. Okay. So that red box here, it's basically the time frame that I am talking about, and you can see that right before vegetation was much more exuberant than it is now. So that it was afterwards, and there were like more temperate trees and so on. But during that period that I am talking about, it is inferred that there were mainly steppe herbs and other herbs. So why did humans go out of Africa? But they did. And they did that between 60 and 40,000 years ago, and now I will present some genetic data that supports that hypothesis. So basically, this is just a cartoon to say in which way we use genetic data. So imagine we have an ancestral population. This is part of the DNA. We track these base pairs, and they can only be A, C, G, or T. And then you can have some individuals that are exactly the same of the ancestral sequence, but then you have some individuals that have mutated. So you can see that individual 1 and 3, they share a mutation, a C mutation, and individual A has another mutation. So you can use this kind of data to infer which individuals are more close to each other than other individuals, which mutations are older. So older mutations will be shared across many individuals and populations, and new mutations will be at low frequencies. And also, we refer to these as mutations, alleles, SNPs, and so on, a bit of nomenclature. So when humans went out of Africa, what happened basically is that there was a great loss of genetic diversity. So just imagine that for like over 100,000 years, humans were in Africa, they were reproducing, new mutations were arising, diversity was generated, and then only a subset of those individuals went out of Africa. So when that happened, of course, only a subset of the genetic diversity went actually out of Africa. And not only it went out of Africa, but it expanded to the entire globe. So the result is that out of Africa populations are much, much less diverse than African populations. So this effect here is called a founder effect. Just a subpopulation going to a new area, it's a founder effect, and that loss of diversity is called a bottleneck. And also this is related with a loss of effective population size. So this is basically like simple population size, but it refers to how many individuals would we need to generate the genetic diversity that we see. So of course, Africa is more diverse, so we need a larger effective population size. Out of Africa, populations are much less diverse, so the effective population size is actually smaller. So we have, luckily, some genetic markers that are witnesses of this out of Africa. They are the mitochondrial DNA and the Y chromosome. So the characteristic of these markers is that they are inherited from mother to daughter and from father to son, respectively. So here we have a family tree, and if we sequence that individual, for instance, we are basically tracking all the mother-only lineages and the father-only lineages. So the downside of this is that you are losing all the genetic diversity that all the other individuals are contributing, but the upside of this is that you can build very reliable phylogenetic trees, so you really can track that marker back in time. Okay, so this is a phylogenetic tree for those who are not familiar with. So let's say that we have an ancestral population and 4 individuals. You can see that those 4 individuals share 2 C mutations. So if we have here the ancestral population as a root, those C mutations are shared across all the other individuals. And then basically by the mutations that are shared, you can build that phylogenetic tree. So what that tells us is that, for instance, individual 1 or 3 are more closely related to each other than they are to individual 2 or 4, for instance. And also you can say, as I mentioned before, that those C mutations are much older, whereas that G mutation is probably much, much younger. Also, some of these mutations are called diagnostic mutations, because imagine, for instance, that here, instead of 2 individuals, we have like 2 subpopulations or 2 groups of individuals. And one group of individuals, they all have a T, and the other group of individuals, they all have an A. We can say that these T and A, they are diagnostic mutations, and then define 2 different haplogroups. And with these haplogroups, studying haplogroups allows us to differentiate populations and make inferences on population growth and so on. So this is the mitochondrial phylogenetic tree. On the bottom line, you have the haplogroups. Don't worry, you don't need to see the letters, it's fine. But what's important is that in green, they are all the Africa haplogroups, and in white, it's the out of Africa haplogroups. So basically, all those mutations that are here, the diagnostic mutations that are here, are shared between all those African individuals that carry that haplogroup and all the out of Africa individuals, and so on. So conceptually, what you can see is that out of Africa populations are actually a subset of African populations. So this would confirm the out of Africa theory. And more particularly, if you focus on this L3 haplogroup, those are the diagnostic mutations shaded in light blue. And again, there are some mutations that are shared by those individuals that are also present in all the out of Africa populations. I would also like you to focus on the fact that there are two haplogroups that I called North African haplogroups, which are called M1 and U6, and we will talk about them later. They are very interesting, and you can already see that they are in the middle of the out of Africa phylogeny, so that's some interesting thing that we will go back later. Oh, and also I would like to add that the length of the branches is proportional to the number of mutations. So actually, African populations have much longer branches because they have accumulated much more mutations than out of Africa populations that, because of the bottleneck, they all had like the same diversity, and then mutations accumulated over a short period of time. So another nice thing about phylogenetic trees and about mitochondrial DNA is that you can calculate how often a mutation fixes in a phylogenetic tree. And this happens more or less at a constant rate. So it's actually possible to calculate how long ago that person that was carrying that haplogroup existed. And basically, for all out of Africa mitochondrial DNA haplogroups, that person existed around 60,000 years ago. So if you have an out of Africa mitochondrial DNA haplogroup, all our, so the women that belong to that haplogroup existed 60,000 years ago. And we all descend from that woman. Of course, does not mean that we all descend from a single woman, but this is just true for that marker, but it's very useful. So we have that around 60,000 years ago, humans went out of Africa. What happened between 40 and 20,000 years ago? From archaeological data, we have the oldest modern human fossils that are found as far back as England, China, and Southeast Asia. They date around 45,000 years ago. So humans expanded, and they did it like quite quick, quite fast. And more interestingly, we have been able to trace that humans went back to Africa, actually, and they settled in North Africa. And I will just now show you the genetic data that we have that shows that. So the basic concept is population structure. We are saying that mutations appear over time. So imagine that there is a single population that for some reason is separated. All the shared mutations from that population, that now are two populations, they will change in frequencies over time, but they will change in different ways depending on stochastic reasons or whatever. But the frequencies of the shared mutations, they will tend to differentiate over time, but also new mutations that arise, they will be different between the two populations. So the longest those populations have been separated, the more genetic differences you can find between them. Of course, if those populations are communicated, the frequencies will tend to get equal because of migration. And also the new mutations will be more likely to be actually shared and be transmitted from one population to the other. Okay, so this is a structure plot. We have several populations that are separated by those white lines. Every bar is an individual. And this is not mitochondrial DNA data anymore. This is genomic data. It's the whole DNA. And it's around 700,000 markers. And we have some African populations, some North African populations, and some Eurasian populations. So basically what I am saying here is that it's a software, it's an algorithm that takes the allele frequencies, and I say, okay, if you were going to split that data set into different ancestries, how they would be distributed on the individual's genome. So here we have two individuals. In this case, orange would mainly belong to African ancestry, and purple would belong to Eurasian ancestry. And here it would be like different proportions of the genome, like 50% of a genome would belong to an African ancestry and the other 50% to Eurasian ancestry. So it's very interesting to notice that North African populations, though they have some amount of African ancestry, they are mainly associated with Eurasian ancestry. So North Africans are more similar to other out of Africa populations than they are to sub-Saharan African populations. This is another plot. This is done with whole genome data, so millions of markers this time. in the x-axis, we have the time in a log scale. So we go from 1 million years ago, 100,000 years ago, and 10,000 years ago. And in the y-axis is the effective population size times 1,000. And I would like to draw your attention to this area here, which corresponds roughly to the time frame that I am talking about. And you can see that those lines here are sub-Saharan African populations. They are hunter-gatherer populations, the San from Namibia and Dumbuti from Tanzania, I think. And those populations are out of Africa populations. The Mozabites that are here in light blue, which are shown here, they have an effective population size that is equal or very, very similar to other out of Africa populations. So if you remember that what I told you before about the effective population size, this basically matches the fact that they first went to that bottleneck when populations went out of Africa, their genetic diversity was reduced, and then they went back to Africa again. Okay, so another interesting thing, this is a structure plot similar to the one that I showed before, but instead of 2 different ancestries, I just put 7 different ancestries. These are other populations that were included in the data set, but for the sake of visualization, I just included North African populations, which are here, and they are oriented from west to east. And the first thing that, well, apart from the interesting fact that there are different amounts of sub-Saharan African ancestry in those North African populations, you see that the North African component exists in all the populations. In this case, this is also North African ancestry, but it's just that the populations that we took were very homogeneous, and that's why that software captured a single ancestry for that population, but take my word for that, they are Maghrebi as well. So it's very interesting here that you have this Maghrebi pattern that basically its distribution is completely opposite to a Near Eastern pattern. So of course this is Egypt, it's really close to the Near East, there's migration. So one question that would be like very interesting to solve is, how long ago did Maghrebis split from Near Easterners and even Europe, which is this purple component that is more like evenly distributed? So in order to do this, we did this analysis. This is the effective population size that we consider it's a constant. We can calculate this, and what we are interested is in this time. So FST is basically a measure of genetic differentiation. It goes from 0 to 1, where 0 would be that the 2 populations that you are analyzing are basically the same. And FST equal 1 would be that they are 2 completely different separate populations. So just to give some reference between humans, the FST values, the maximum FST value that you can get, it's between 0.2, maybe 0.25 or something like that. And I would like you to focus on the points right now. So these are the FSTs, and then this is the time basically that we get from that equation here. So the points are basically comparing populations. And then all the purple points are comparing every North African populations that we have with the Basques, actually, in the Iberian Peninsula. And the green points are the same North African populations compared with the Near Eastern populations that we have that are the Qatari from the Arabian Peninsula. So as you can see, well, first, it's interesting to see that those populations split prior to the Holocene. So this is already a very interesting result in the field, and I will explain afterwards why. But we wanted to go a bit, we wanted to push the data a bit with more accuracy, because of course we have seen that the Egyptian population actually have a lot of this Near Eastern ancestry. So when you calculate the genetic differentiation, you are taking this into account. So you are actually losing all the possible genetic diversity. So what you can do instead is, instead of comparing or calculating the genetic differentiation between the populations, we wanted to do it between the components. So forget about the populations, just calculate it between this light blue component, the green component, and the light blue component, and the purple component, which would be the European. And these are the shaded areas. So when when you are able to ignore the admixture, the migration that has been between populations, those dates go much further back. And they are like, you can conclude that the Maghrebi split from the Near Eastern at least 20,000 years ago. And this was probably actually much larger. We have an upper bound of 40,000 years ago. Okay, so this is the U6 haplogroup, is the one that I told you to remember, that it's actually North African, but it's in the middle of the out of Africa phylogeny. Here you have the distribution of the haplogroup. You can see some very low frequencies in Asia and East Africa, and rather high frequencies in Western North Africa. But I say rather, like they reach 10% frequency, and it's the place where they reach the highest frequency. So they are quite find at low frequencies. But it's very interesting because if you look at how long ago did that haplogroup originated, it was 40,000 years ago. So we have an haplogroup 40,000 years ago. If you do the phylogeny, the root is around here, and then it went west. So this is actually a proof also of that back to Africa wave. And it's dated around 40,000 years ago, which is actually also similar to our result. So to finish that part, just to remember the heading, like 40,000 years ago, more or less, modern humans went back to Africa and settled there. We are getting closer. What happened between 20 and 10,000 years ago? So there is the last glacial maximum, the post-last glacial maximum, and what we have is that North Africans expanded after the last glacial maximum. I would have put the African humid period if I was more aware about climatology. So we can know this doing haplogroup networks. So this is very similar to the phylogeny. This is the same individuals that I showed you before. Remember the sharing Cs and the As. So basically, if this was the ancestral individual that we were talking about, we would have a CC change. We do not have that individual, but we assume it existed at some point. And then two individuals obtained an A, like a new mutation that was an A. Note, please, that the circle is double. So in those haplogroup networks, the size of the circle is actually important. It means how many individuals have the same haplogroup, and so on. So again, we have the U6 haplogroup. This is a phylogenetic network. In red, there are like the mutation positions that go from one place to another. But what it's the most interesting part here is that green is in Maghreb. So basically, the frequency of the presence of this haplogroup is in Western North Africa. But more importantly, you find those big circles and then all small circles that branch off from the node. We call this the node. So this is called a star-like shape distribution. And it's actually in agreement with population expansion. So if you just imagine at some point individuals start to expand, to reproduce more, that haplogroup will prevail over time, and then you will have more and more individuals that belong to that haplogroup. After a certain period of time, some of those individuals will mutate, and then you will find new mutations and new haplogroups branching from that one. But you don't have like a stepwise thing that one haplogroup, one mutation, another haplogroup, but it's a radial thing. So that's a signal of a population expansion. And if we date how long ago did that happen, it's around 13,000 years ago, which I guess it matches some of the evidences of the onset of the African humid period. And we can discuss that later. What about haplogroup M1? That haplogroup is much more frequent in Egypt, not in Western North Africa, and it shows a similar sign of expansion. And again, if you date M1A1, which is basically that haplogroup that is more present in Egypt, you find the same results. Like 13,000 years ago, there was a population expansion. So this is not only true through genetic data. If you look at the archaeological data, you also find some drastic differences, like changes in subsistence strategies. Apparently, 13,000 years ago, though the technology, the lithic technology that existed has not changed, you do see a change in lifestyle. And you start to see a lot of terrestrial snail shells remains. And that gives rise to the hypothesis that there was a more change in subsistence strategies, like more, less hunting and more gathering, and also a more sedentary lifestyle. And this is in Western North Africa. But in Eastern North Africa, you also find a similar thing. So there are hypotheses that the Nile River Valley was actually last glacial maximum refugia. And because wildlife just gathered there, there was a more sedentary lifestyle because resources were very abundant. And even it has been suggested that there were evidences of proto-agriculture, though most of the studies say that it was actually a cul-de-sac. So we have that after the last glacial maximum, North Africans expanded. So let's move to the last 10,000 years. This is the Neolithic. It's very controversial and very interesting. So during the 1980s, there was the most accepted theory about how the Neolithic spread in Europe and North Africa was that it appeared in the Near East, and then it went from the Near East to Egypt, then it probably stayed there for a while, and then it spread over both shores of the Mediterranean in a parallel way. And well, so one of the main questions was, okay, that spread was mainly cultural, or was actually a population replacement, where actually people from the Near East entering to North Africa and replacing the extant population. Also, I would like to say, in Western North Africa, there are more evidences of pastoralism than agriculture, which is also very interesting. So the Neolithic evidences that we have from North Africans are on the Y chromosome. The system is very similar to the one that I explained for the mitochondrial DNA. This is a phylogenetic tree of the Y chromosome. And you can see that most of the North African populations have very, very high frequencies of a specific haplogroup, which is called E3b, and also quite high frequencies of another haplogroup, which is E3b2, sorry. And they are very, very close in the phylogeny, so they are directly related. So if you date the phylogeny of these two haplogroups, it's very recent. It's 8,000 and 4,000 years ago. So at the beginning, I thought that that was very surprising because the coalescence, like the time of the most recent common ancestor of the mitochondrial DNA is, for the North African haplogroups of the mitochondrial DNA, is around 40,000 years ago. And whole genome data shows us that the Maghrebi component, as I showed before, split from the Near Eastern one, also far back in time, like between 20 and 40,000 years ago, but the Y chromosome is 8,000 years ago. So what could explain that? What could explain that only those lineages that are very frequent in North Africa and are present in so many populations have only 8,000 years ago? So, well, I call this like the Genghis Khan effect, sorry for the mispronunciation. So this is basically when a group of males carrying the same haplogroup, or in this case, a single male carrying this haplogroup, is very successful reproductively. So apparently, Genghis Khan was very, very successful during his expansion. So he was carrying this haplogroup here. The colors mean different geographic areas. Not all of them are in China. They go as far as Western Asia, Siberia, and they are all over. And they are all carrying the same haplogroup. So this would be explained by him being very prolific, having a lot of progeny with the same haplogroup, that progeny having other progeny, and so on. So what you can see is that star-like shape of population expansion that I was talking about. So it is possible, going back to our North Africans, is that some males, for some reason, were highly successful at that period and left a lot of progeny, replacing other haplogroups. So the question that we have not been able to answer is that whether that was due to the Neolithic to the fact that they were able to produce more food resources and so on. They were more successful and they spread all over. And I think that this is something that it's worth studying. Another nice story about the Neolithic is that North Africans become lactose tolerant. So this is an example I just want to show you, which is the genetic footprint that a very beneficial mutation leaves on the DNA. So imagine that you have a population. As I said, over time there is variation in every individual. Some variation is shared, some variation is unique to the individual, and so on. But then a beneficial mutation that is shown here in red arises. So if this mutation is beneficial enough, what will happen is that that individual will have more chances to reproduce than other individuals. So, and if that really makes a difference, in a short period of time, what you will see is that not only a lot of individuals carry the beneficial mutation, that is what you would expect, but also the other variation that was associated to that individual and that is actually found at very low frequencies in the population. Now it's found at very high frequencies. And we call this a selective sweep. So what I did was, we have some genome data from a population in Algeria, and I just went and looked at the position of the lactase gene, and here every line is a haplotype. This is the region of the lactase gene. It's worth to mention that actually the mutation that affects the lactase gene is somewhere here before the gene, and it just affects it and then allows you to digest milk, which there's no need to mention why this is beneficial. And what you can see is a very, very nice selective sweep. So all this variation is actually, we say that it's linked to the beneficial mutation. And also it was nice to see that probably there is an individual here that is not able to digest milk, just for the record. So we have this information here. So basically, what we see is that North Africans, probably present-day North Africans, they came to North Africa around 40,000 years ago or so. They underwent a population expansion right after the last glacial maximum. Unfortunately, we do not have the kind of data that we need to evaluate whether there was a population expansion or not during the Neolithic. We have not been able to track it, at least. But also, I would like to add some archaeological evidences that just to try to discuss a little bit that theory about a continuum wave of Neolithization from Egypt to Western North Africa. So the Neolithic was established around 7,500 years ago. It's most likely that it had a Near Eastern origin, and it changed the lifestyle of the populations. There are a lot of evidences of changes in lifestyle. But in Western North Africa, actually in Morocco, the first evidences of agriculture date around 6,000 years ago, but there is actually a 3,000 years gap between the first evidences of pottery and the first evidences of agriculture. So usually people call a Neolithic package, which is basically when Neolithic arrived, people started to use pottery, to raise crops, and to become pastoralists, and also to change from a nomad lifestyle to a sedentary lifestyle. So we have evidences in North Africa that sedentary lifestyle actually appeared before the Neolithic. We also have this use of pottery that is unlinked to the pastoralism. So what is Neolithic? Like, what was first? Or what was the influence actually from the Near East that we were talking about? And these are questions that are kind of difficult to answer. And to make things even more difficult, in East Algeria and Tunisia, which is basically in the middle between Egypt and Western North Africa, we have hunter-gatherers while all the rest of the people were Neolithic. So this is also something that is kind of confusing, and that I guess that it's worth discussing. So just to finalize, to conclude, which is the genetic evidences that we have. So we have shown that some North African males left more offspring than others, replacing almost a complete Y chromosome pool. So why did that happen? Were those people pastoralists or not? I think that a very interesting thing to do would be to compare the Y chromosome lineage from Western North Africa and from Tunisia, or from the places where theoretically hunter-gatherers outlasted, and see if there are differences. So that could give us some answers regarding that, and as far as I know, nobody has done it yet. Also, it is clear that individuals who were able to digest milk left more offspring, and then the mutated gene spread fast in the region. There is a clear genetic footprint that we can detect on that. And also, present-day North Africans did not come with a Neolithic from the Near East, but adopted it culturally. So the question that remains is whether they adopted it culturally from the Near East or from other Neolithic source, which is also open for discussion. And in agreement of archaeological data, that adoption was mainly not genetic, but cultural. And with that, I would like to acknowledge my former thesis supervisor, David Comas, and also Carlos Bustamante and Berna Hen, which at the time were collaborators. And Berna Hen is actually now my PI, and I am her postdoc. And I would like to thank you for your attention.